So let's continue with the gastric motility. Gastric motility refers to the coordinated contraction and relaxation of the muscles in the stomach, which allows for the mixing and breakdown of ingested food. The procedure for gastric motility involves multiple steps and it is regulated by the enteric nerve system and the autonomic nerve system. The process of gastric motility can be divided into two phases, the fasting phase and the fed phase. Fasting phase, during the fasting phase, the stomach is relatively empty and the muscles in the stomach are relaxed. The relaxation of the muscle allows for storage of ingested food and the secretion of gastric juice. The migrating motor complex, MMC, a series of coordinated contractions that originate in the stomach and move down the small intestine, helps to clear any residual food or debris from the stomach during this phase. Next, fed phase. During the fed phase, food enters the stomach, triggering a series of contractions known as mixing waves. These contractions mix the food with gastric juice and break it down into smaller particles. The muscles in the stomach also work to grind the food into the semi-solid mass known as chyme. The chyme is then gradually emptied into the small intestine through the pyloric sphincter. The nerves control of gastric motility is complex and involves multiple pathways. The enteric nerve system, which is a network of neurons that lines the digestive tract, plays a key role in regulating gastric motility. The enteric nerve system is divided into two main plexus, the myenteric plexus and the submucosal plexus. The myenteric plexus is responsible for regulating the contraction and relaxation of the muscles in the gastrointestinal tract, including the muscles in the stomach. The submucosal plexus regulates the secretion of gastric juice and hormones that affect gastric motility. The autonomic nerve system, which is controlled by the brain and spinal cord, also plays a role in the regulating gastric motility. The sympathetic nerve system, which is responsible for the body's fight or flight response, inhibits gastric motility. The parasympathetic nerve system, which is responsible for the body's rest and digest response, stimulates gastric motility. So, a sympathetic nerve system is something which is responsible for the body's flight or fight response, which inhibits gastric motility, but the parasympathetic system stimulates gastric motility. The muscle associated with gastric motility include the circular muscle layer and the longitudinal muscle layer. The circular muscle layer, which is the innermost layer of the stomach muscles, contract to mix food with gastric juice and break it down into smaller particles. The longitudinal muscle layer, which is the outermost layer of the stomach muscle, contracts to move food along the length of the stomach and into the small intestine. In summary, gastric motility is a complex process that involves the coordinated contraction and relaxation of the muscles in the stomach. The process can be divided into two phases, the fasting phase and the fed phase. Gastric motility is regulated by the enteric nerve system and the autonomic nerve system, and it involves multiple pathways and muscles. Proper gastric motility is essential for the efficient breakdown and absorption of nutrients from ingested food. Gastric emptying. In detail, gastric emptying refers to the process by which food and liquid that have been broken down in the stomach are released into the small intestine for further digestion and absorption. This process is essential for the efficient digestion and absorption of nutrients from ingested food. The procedure for gastric emptying involves multiple steps. What is happening in this gastric emptying? Contents of the stomach are released into the small intestine for further digestion and absorption. The major points involved in gastric emptying include the following: release of chyme. Once the food is broken down into a semi-solid mass called chyme, it is released from stomach into the small intestine through the pyloric sphincter. Then rate of gastric emptying. The rate at which food is released from the stomach into the small intestine is regulated by various factors such as the composition and volume of the meal, the presence of hormones and neurotransmitters, and the tone of the pyloric sphincter. The rate of gastric emptying is the rate at which food is released from the stomach into the small intestine is regulated by various factors such as the composition and volume of the meal, the presence of hormones and neurotransmitters, and the tone of the pyloric sphincter. Then effect on digestion and absorption. The rate of gastric emptying can have a significant impact on digestion and absorption. Rapid gastric emptying can lead to incomplete digestion and malabsorption, while delayed gastric emptying can result in overloading of the small intestine and impaired absorption. The procedure of gastric emptying involves multiple steps, including the mixing and breakdown of ingested food in the stomach, the regulation of the rate of gastric emptying, and the release of chyme into the small intestine. The following factors play a significant role in regulating gastric emptying. Hormones such as gastrin, cholecystokinin, and secretin are released in response to ingested food and help to regulate the rate of gastric emptying. Autonomic nerve system. The autonomic nerve system, which is responsible for the body's rest and digest response, stimulates gastric motility and promote gastric emptying. Presence of food. The presence of food in the small intestine stimulates the release of hormones and neurotransmitters that help to regulate gastric emptying. Then volume and composition of meal. The volume and composition of the meal can also affect the rate of gastric emptying. Fatty meals and meals high in protein are known to delay gastric emptying, while meals high in carbohydrate tend to stimulate gastric emptying. Gastric emptying is an important process that allows for the sufficient and efficient digestion and absorption of nutrients from ingested food. It is regulated by various factors, including hormones, the autonomic nervous system, and the volume and composition of the meal. Proper regulation of gastric emptying is essential for maintaining optimum digestive health and preventing complications such as malabsorption and nutrient deficiencies. Next, gastric emptying process. Gastric emptying is a complex process that involves the coordination of multiple nerves and muscular components. The procedure can be divided into several stages, like mixing of food. When food is ingested, it is mixed with gastric secretions and turned into a semi-solid mass called chyme. Then regulation of gastric emptying. The rate of gastric emptying is regulated by various factors, including the composition and volume of the meal, the presence of hormones and neurotransmitters, and the tone of the pyloric sphincter. Then contraction of gastric muscles. Once the pyloric sphincter opens, the gastric muscle contracts to propel the chyme into the small intestine. These contractions are regulated by the enteric nerve system, which is a network of neurons within the gastrointestinal tract that is capable of regulating digestive function independently of the central nervous system. Then release of chyme. The chyme is released into the small intestine through the pyloric sphincter. The small intestine then continues the process of digestion and absorption of nutrients. The nervous control of gastric emptying involves several components, including the vagus nerve, which is responsible for stimulating gastric motility and increasing the tone of the pyloric sphincter. The enteric nerve system, which is located within the wall of the gastrointestinal tract, is responsible for regulating the contractions of the gastric muscles and coordinating the release of chyme into the small intestine. The muscles involved in gastric emptying include the smooth muscles of the stomach, which contract to mix and break down the ingested food, and the pyloric sphincter, which controls the release of chyme into the small intestine. The contractions of these muscles are regulated by the enteric nerve system and are influenced by various factors, such as the volume and composition of the meal, the presence of hormones and neurotransmitters, and the tone of the pyloric sphincter. In summary, gastric emptying is a complex process that involves the mixing and breakdown of ingested food, the regulation of the rate of gastric emptying, the contraction of gastric muscles, and the release of chyme into the small intestine. The procedure is regulated by various factors such as hormones and autonomic nerve system and the enteric nerve system. The proper regulation of gastric emptying is essential for maintaining optimal digestive health and preventing complications such as malabsorption and nutrient deficiencies. Intestinal motility in detail with explanation and major points. Procedure. Intestinal motility refers to the movements of the contents of the small and large intestines through the contractions of the intestinal smooth muscles. The process of intestinal motility is important for digestion and absorption of nutrients, as well as for the elimination of waste products. The procedure of intestinal motility can be divided into several stages, like segmentation. The intestine contracts rhythmically in small segments, which mix the contents of the intestine with digestive juices and increase the surface area for absorption of nutrients. Then peristalsis. Peristalsis is a coordinated contraction of the smooth muscle that propels the contents of the intestine forward. The circular muscle behind the bolus or foot mass contract, while the muscles in front of it relax, pushing the bolus forward. Defecation. The final stage of intestinal motility is defecation, where the waste products are eliminated from the body. This process is regulated by the reflexive relaxation of the internal anal sphincter and voluntary relaxation of the external anal sphincter. The regulation of intestinal motility is a complex process that involves both the enteric and autonomic nerve system. The enteric nerve system, which is composed of neurons located within the walls of the intestine, regulates the local reflexes that control intestinal motility. The autonomic nerve system, on the other hand, regulates the overall coordination of intestinal motility and is influenced by various factors such as emotions, stress, and presence of food. The importance of proper intestinal motility cannot be overstated. When the mobility is too slow, food may be retained in the intestine for too long, leading to constipation and potentially harmful bacterial growth. Conversely, when motility is too fast, food may pass through the intestine too quickly, leading to malabsorption and nutrient deficiencies. Examples of conditions that can affect intestinal motility include irritable bowel syndrome, inflammatory bowel disease, and neurological disorders such as Parkinson's disease. Treatment options for these conditions often focus on regulating intestinal motility through the use of medications and dietary modifications. In summary, intestinal motility is a complex process that involves the segmentation, peristalsis, and defecation of the contents of the small and large intestine. The process is regulated by both the enteric and autonomic nerve system and is important for proper digestion, absorption, and elimination of waste products. Understanding the mechanisms of intestinal motility is essential for diagnosing and treatment of gastrointestinal disorders. Next, intestinal motility procedure. The procedure of intestinal motility involves the coordinated contraction of the smooth muscles of the small and large intestine. These contractions are regulated by a complex interplay of nerves and hormonal signals and involve a variety of different muscle types. The smooth muscle cells of the intestine are arranged in two layers: the circular muscle layer, which surrounds the intestine, and the longitudinal muscle layer, which runs parallel to the length of the intestine. These two layers work together to produce peristaltic movements of waves of contractions that propel the contents of the intestine forward. The process of intestinal motility is regulated by both the enteric nerve system and the autonomic nerve system. The enteric nerve system is a network of neurons that is embedded within the walls of the intestine. These neurons are responsible for coordinating the local reflexes that control intestinal motility, including the release of neurotransmitters that stimulate or inhibit smooth muscle contraction. The autonomic nerve system, on the other hand, regulates the overall coordination of intestinal motility. It consists of two branches: the sympathetic nerve system, which inhibits intestinal motility, and the parasympathetic nerve system, which stimulates it. The muscles associated with intestinal motility include the circular and longitudinal muscle layers of the intestine, as well as the sphincter muscles that control the movement of material through the intestine. The circular muscles are responsible for the segmentation of the intestine, while the longitudinal muscles work in concert with the circular muscles to produce peristaltic waves. In addition, there are several sphincter muscles that regulate the movement of material through the intestine. This include the ileocecal valve, which regulates the flow of material from the small intestine into the large intestine, and the internal and external anal sphincters, which control the elimination of waste products from the body. And disorders of intestinal motility can lead to a variety of gastrointestinal symptoms, including constipation, diarrhea, and abdominal pain. Treatment options for these disorders may include medications that stimulate or inhibit intestinal motility, dietary modifications, and surgical interventions. In summary, intestinal motility is a complex process that involves the coordinated contraction of the circular and longitudinal muscle layers of the intestine, as well as the regulation of sphincter muscles that control the movement of material through the intestine. The process is regulated by both the enteric and autonomic nervous system and is important for proper digestion, absorption, and elimination of waste products. Understanding the mechanisms of intestinal motility is essential for the diagnostic purpose and treatment of gastrointestinal disorders. Then defecation process. Defecation is the process of elimination of feces from the body. It is a vital part of the digestive process. It involves the coordinated action of several muscles and nerves in the digestive system. The major points involved in the defecation process include the relaxation of the sphincter, internal anal sphincter, the contraction of the rectum, and the relaxation of the external anal sphincter. The procedure begins with the filling of the rectum with feces. As the rectum fills, it stretches, which triggers a reflex called the rectosphincteric reflex. This reflex causes the internal anal sphincter to relax, allowing feces to move into the anal canal. Next, the rectum contracts, which increases the pressure within the rectum and pushes the feces towards the anal canal. At the same time, the external anal sphincter relaxes, allowing the feces to pass out of the body. This process is under the control of the nerve system, specifically the parasympathetic nerve system, which activates the defecation reflex. The defecation process is important for the elimination of waste products from the body. If feces is not eliminated regularly, it can accumulate in the colon, leading to constipation, which can cause discomfort, pain, and other health problems. There are several factors which can affect the defecation process. This include diet, hydration, and physical activity. A diet high in fiber can promote regular bowel movements, while dehydration and sedentary lifestyle can contribute to constipation. Defecation is a complex process that involves the coordinated action of several muscles and nerves in the digestive system. The process is important for the elimination of waste products from the body and can be influenced by factors such as diet, hydration, and physical activity. Understanding the mechanisms of defecation is essential for maintaining proper digestive health and preventing complications such as constipation. The process of defecation involves a coordinated action of several muscles and nerves in the digestive system. The major steps in the defecation process include a rectal filling, rectosphincteric reflex, rectal contraction, and relaxation of the external anal sphincter. Rectal filling. The rectum serves as a storage area for feces until they can be eliminated from the body. As the rectum fills with feces, it stretches and triggers the rectosphincteric reflex. This reflex is initiated by the stretched receptors in the rectal wall, which send signals to the spinal cord. Rectosphincteric reflex. The rectosphincteric reflex involves the coordinated action of the internal anal sphincter and the rectum. The internal anal sphincter is a smooth muscle ring that surrounds the anal canal and is under involuntary control. When the rectum is stretched, the reflex causes the internal anal sphincter to relax, allowing feces to move into the anal canal. This reflex is controlled by the parasympathetic nervous system. Rectal contraction. Once the feces have moved into the anal canal, the rectum contracts to increase the pressure within the rectum and push the feces towards the anal canal. Rectal contraction is controlled by the enteric nerve system, which is a complex network of nerves that is responsible for regulating the functions of the digestive system. Then relaxation of the external anal sphincter. The external anal sphincter is a skeletal muscle ring that surrounds the internal anal sphincter and is under voluntary control. And the rectal contraction increases the pressure within the anal canal. The EAS must relax to allow the feces to pass out of the body. The relaxation of the EAS is controlled by the somatic nerve system, especially the pudendal nerve. Then muscles associated with defecation. Internal anal sphincter. The internal anal sphincter is a smooth muscle ring that surrounds the anal canal and is under involuntary control. It is responsible for maintaining anal tone and preventing involuntary leakage of feces. Then external anal sphincter. The external anal sphincter is a skeletal muscle ring that surrounds the internal anal sphincter and is under voluntary control. It allows for the control of the defecation and prevents the involuntary leakage of feces. Then rectal muscles. The rectal muscles are responsible for the contraction of the rectum during the defecation process. These muscles are controlled by the enteric nerve system. Then nerves control. The defecation process is controlled by the nervous system, specifically the parasympathetic nerve system, enteric nerve system, and the somatic nerve system. The parasympathetic nerve system activates the rectosphincteric reflex, which causes the IAS to relax. The enteric nerve system controls the contraction of the rectal muscle, and somatic nerve system controls the relaxation of the EAS. The process of defecation involves a coordinated action of several muscles and nerves in the digestive system. The defecation process is controlled by the parasympathetic nerve system, enteric nerve system, and the somatic nerve system. Understanding the mechanisms of defecation is essential for maintaining proper digestive health and preventing complications such as constipation. The role of hormones in the control of gastrointestinal motility. Hormones play an important role in regulating the gastrointestinal motility, which is the movement of food through the digestive tract. These hormones are produced by various endocrine glands located throughout the body and act on the smooth muscles of the GI tract to regulate their contraction and relaxation. Here are some hormones and their role in controlling GI motility. First one is the gastrin. Gastrin is a hormone produced by the G cells in the stomach. It stimulates the secretion of gastric acid and pepsinogen, which help in the digestion of food. Gastrin also promotes gastric motility by increasing the contraction of stomach muscles. Cholecystokinin or CCK. CCK is a hormone produced by the duodenal mucosa in response to the presence of fat and protein in the small intestine. It stimulates the secretion of pancreatic enzymes and bile from the liver and gallbladder respectively. So, CCK is produced from the duodenal mucosa because the small intestine and chyme contain fat and protein. Then that stimulates the secretion of pancreatic enzymes and bile from the liver and gallbladder. CCK also inhibits gastric motility, which slows down emptying of the stomach. Gastrin promotes gastric motility, but CCK inhibits the gastric motility. Then secretin. Secretin is a hormone produced by the duodenal mucosa in response to the presence of acidic chyme in the small intestine. It stimulates the secretion of bicarbonate from the pancreas and inhibits the secretion of gastric acid. Secretin also slows down gastric motility, which allows some time for the pancreatic enzymes to digest the food. Then motilin. Motilin is a hormone produced by the duodenal mucosa. Once again, just read the secretin portion. Secretin is a hormone produced by the duodenal mucosa in response to the presence of acidic chyme in the small intestine. It stimulates the secretion of the bicarbonate from the pancreas and inhibits the secretion of gastric acid. Secretin also slows down gastric motility, which allows more time for the pancreatic enzymes to digest the food. Then motilin. Motilin is a hormone produced by the duodenal mucosa that stimulates the contraction of the smooth muscles in the stomach and small intestine. It is responsible for the migrating motor complex, which is a series of contractions that occur every 90 to 120 minutes during fasting to clear any residual fluid from the GI tract. Then Ghrelin. Ghrelin is a hormone produced by the stomach that stimulates appetite and promote food intake. It also increases gastric motility and acid secretion to prepare the stomach for the incoming food. The procedure of hormone regulation of GI motility include the release of hormones in response to various stimuli such as the presence of food, acidity, and stretching of stomach and intestine. These hormones then bind to the specific receptors in the stomach muscle cells of the GI tract, which causes a cascade of extracellular events that ultimately lead to the contraction or relaxation of the muscles. The nervous control of GI motility involves the enteric nerve system, which is a complex network of neurons located within the walls of the GI tract. The system can function independently of the central nervous system and can regulate the contraction and relaxation of the smooth muscles in response to various stimuli such as stretch, acidity, and hormones. The enteric nerve system receives input from both sympathetic and parasympathetic nervous system, which can modulate the activity of the GI tract. In conclusion, hormones play a crucial role in regulating the GI motility by stimulating or inhibiting the contraction of the smooth muscles in the stomach and intestine. The procedure involves the release of hormones in relation to various stimuli, which then bind to specific receptors in the smooth muscle cells. The nervous control of GI motility involves the enteric nerve system, which receives input from both the parasympathetic and sympathetic nerve system, The understanding of these mechanisms is important for the diagnostic purpose and treatment of various GI disorders.